Hello everyone, and welcome to our online lecture series for Bio1. Today, we cover another one of those flagship topics, something that you can definitely expect to be able to draw come final time, and something that really combines all of your understanding from the first portion of the class where we talked about cells and plasma membranes with our understanding of organismal biology and organ systems. So today, we tackle the broad topic of osmoregulation in the body. Osmoregulation is, of course, balancing our water. And while it may be kind of counterintuitive, or the only way to balance our water is by drinking more of it, you can actually do a lot of balancing of water by balancing the electrolytes in the water. And one of the reasons why it's so important to balance our electrolytes is that our cells really need a perfect concentration of dissolved things to water in order to maintain their appropriate shape. In an isotonic solution, as seen here, you will have our red blood cells floating around, regularly expanded, maintaining their biconcave shape, performing that oxygen and carbon dioxide exchange the most efficiently because of their maximized surface area. However, if things get tilted so that there is a higher abundance of water floating around the red blood cells, meaning the electrolytes are down in the bloodstream, you will encounter a hypotonic solution, wherein now water will rush into the cell to try to compensate. On the other side, if there is too many electrolytes in the bloodstream, too many dissolved things, you will encounter a hypertonic solution. Now, of course, both the hypertonic and hypotonic solutions will result in cell death, either by explosion or by the crenation that begins to fold in the plasma membrane to the point where it is no longer sufficiently or structurally sound so that it breaks down. So when we're talking about the electrolytes that are present in our bloodstream, we're of course talking about dissolved ions or molecules. Because water dissolves many things. Now, based on their electronegativity. Obviously not lipids, right? Again, hitting on some of those early class topics. These ions or molecules then have certain transport proteins, whether they be channels or uniporters or symporters, that move them across our semipermeable membrane. They all need to be able to pass the plasma membrane. And so therefore, this osmoregulation is going to rely heavily on which transport proteins are present at the surface, what they can let in and can let out, and which way diffusion is forcing them to go. And that is probably the most important topic to carry into all of this. With our semipermeable membrane, I'll do a little quick illustration here. This is the inside, this is the outside. Because that membrane, that lipid, prevents things from crossing, even if you had 99 particles of sodium inside and 1 particle of sodium outside, without some sort of transport protein to allow those things to migrate across, you would get very, very little diffusion across the plasma membrane. The sodium would not be leaving. This is the same principle that we saw in our neurons, right? Our resting state was a case where there was a high concentration of one ion on the inside and another high concentration of a different ion on the outside. And because the transport proteins were not there, you maintained this resting state balance. Now, of course, the entire idea of a nerve impulse was based on the fact that you could stimulate the opening of those transport proteins. In the case of osmoregulation, we are instead going to rely on hormones to move transport proteins to the cell surface. Whoop, that's not supposed to be there. Or open those that are already present. So you can go either way. And of course, we have that example of the GLUT4 receptor being moved to the plasma membrane of our red blood cells in the case of the hormone insulin, signaling to these molecules, to these red blood cells rather, that they need to move to the plasma membrane so that way they can hide glucose within themselves. The ions that we're going to talk about today are sodium with a positive charge, potassium with a positive charge, calcium with a 2 positive charge, and magnesium with a 2 positive charge. We also have several negative ions, chlorine with a single negative charge, carbonate, CO3, 2 minus, HCO3 minus, bicarbonate. This, of course, is our buffer that is present when CO2 is dissolved in the blood. You definitely want to manage that for pH reasons. And then phosphite ions, PO3 minus, which are part of the readily added and readily removed ATPs. So those ions are important things to keep track of. They're going to be the main focus of the ions, the filtrate that we're talking of, the ions removed from the filtrate or added back into the filtrate, with the filtrate itself being the liquid that is being filtered on its way to becoming substance in your urinary bladder. So it's the pre-PP. And it is true that the vast majority of the electrolytes that you lose, electrolyte loss, occurs during excretion and perspiration, or sweating. So you do lose a lot of electrolytes through those two means. The organ that is responsible, or organs rather, that is responsible for governing this process of excretion are your ever important kidneys. They are the filtration organ. They have one quite very interesting statistic, is that 1/4 of all the oxygen you breathe is used up by your kidney, because they are so metabolically intensive in their energy use for filtration, moving transport proteins, opening transport proteins, closing transport proteins, filtering things out. It is so much work and requires so much cellular respiration that your kidneys themselves are using up 1/4 of the air that you breathe. That's nutty to me. Of course, your kidneys are found near the spine, in your back. You have 2 kidneys each, but only one is needed to maintain. One is sufficient to be able to filter the blood for an individual. So this is why people can donate kidneys, and it is life without a kidney is extremely painful. The process of dialysis is no fun, and having to do it on the regular is of course both expensive and very difficult, physically and mentally. Yeah, do take care of your kidneys. Now, as to the structure and important features of the kidney that I expect y'all to be familiar with. Here, I've left blanked out all the terms and all the regions that I hope you keep track of. Of course, you're familiar with the capillaries, the blood-carrying vessels that are present in the kidney that will ultimately lead to the glomerulus. You have arterioles and venules, the arteries and veins that carry blood in and carry blood out. The major and minor calyxes, which are essentially just cups in this massive feature called the renal pelvis. More on this structure in a moment. And then we have a medulla, which is a particular region of the kidney with nephrons, and the cortex, the region on the outside, so these two structures. And then we have the fascia and capsule, the features that actually make the boundary of the kidney. So, as to the things that you need to know, the first is the renal pelvis, this giant collecting duct. For the filtrate, it is actually a soft tissue, so it's kind of like a blood vessel in that it is a soft, flexible tissue, but rather than carrying blood, it is always just carrying processed filtrate. So this is your urine on your way to your urinary bladder. So the renal pelvis has many different cups, or these calyxes, that then feed into the ureter, or the transport tube for your urine on its way to the urinary bladder. The functional units of the kidney are renal pyramids. So individual renal pyramids are stacks of nephrons, one on top of the other. More on those in a sec. And then we have our two blood-carrying vessels, the renal vein and the renal artery. The renal vein will lead directly to the inferior vena cava. So those of you who are familiar with anatomy may know that the vena cava is your large vein cavity. Again, kind of similar to that renal pelvis in that they are just giant places where things accumulate. The vena cava obviously carrying blood, not urine, but and in this case operating against gravity because you need to travel up from the kidneys to the heart. Excuse me. And then the renal artery carrying highly oxygenated blood to the kidneys. In terms of a smaller structure, so looking at each individual renal pyramid itself, so here we have the entire stack of many different nephrons, the nephron being the functional unit for a kidney. And we see nephrons in incredibly tiny organisms. Earthworms and other invertebrates have nephridia, which are essentially analogous structures, even though they evolved independently. So this feature of filtering out ions that you do not need or do not want in your bloodstream, or are wastes, is something that is consistent across all animals. Our nephrons are structured a little bit differently, and so we'll be paying attention to the mammalian nephron type. Now, the structures of the nephron itself, again, just like I had above here, where I should highlight the terms that I expect y'all to be familiar with: the renal vein, renal artery, the ureter, the renal pelvis, and a singular renal pyramid. I have structures here that I expect you to be familiar with: the nephron itself, our renal pyramid, and then these individual components. But before we get to those, let's talk a little bit about the nephron itself. There are two different types of nephrons: cortical nephrons that make up 85% of your nephrons. These are ones that are present in the cortex. And then you have ones that are juxtamedullary. And these are about 15% of your total nephron amount. So most of them are these cortical nephrons present primarily in the cortex. The nephron is the functional unit of the kidney. And you've got just over 1 million nephrons per kidney. So that's over 2 million nephrons per human. The nephron itself has a functional unit called the renal corpuscle. Or the renal body, and its capillaries. And this is the blood filtrate interface. So this is actually where you're going to squeeze the blood to remove everything that is present in there, and then afterwards grab back the pieces that you want. And that's the way that this nephron works. The idea is that there is a bag, the glomerulus, and a Bowman's capsule, these two making up your renal corpuscle, wherein blood will flow in from the efferent arteriole, from the afferent arteriole, so afferent bring in, efferent bring out. And when the blood goes into this glomerulus, which is a bag of endothelial tissue, so this is our soft connective tissue, the Bowman's capsule actually provides a stiff outside. So it squeezes that blood kind of like a sponge. And just like when you squeeze a sponge, none of the red blood cells should be going through. But when you squeeze that sponge, the liquid and everything that's attached to it, the soap, the grime, the dirt, it all squeezes out. And that Bowman's capsule actually catches all of those components. The components that go into the Bowman's capsule is called our filtrate. After the filtrate leaves the Bowman's capsule, it goes into the proximal convoluted tube, and then, let's see if I can grab a highlighter to illustrate this. It'll be filtered here in the Bowman's capsule glomerulus, the renal corpuscle. It then gets transported along this proximal convoluted tube, and proximal means close side. It then goes down into the descending loop of Henle before coming back up the ascending loop of Henle, and then it goes into our distal convoluted tube before adjoining to our collecting duct. And the collecting duct, of course, goes on to your ureter. So, glomerulus, term you need to know. Bowman's capsule, term you need to know. The glomerulus is actually holding the blood. The Bowman's capsule is holding the filtrate. The proximal convoluted tubule is the first tube that the filtrate's going through. We'll talk about what happens there in a second, but this is again on the way to becoming urea. Then down the loop of Henle, of which there are two sides, a descending loop and an ascending loop that are cellularly very different. and then the distal convoluted tube. Now, each of these convoluted tubes are named such because they twist and turn a lot to maximize the surface area for cells to bring filtrate back in. So there's a reason that they are convoluted. And then proximal means close, distal means far. And so this is in relation to the glomerulus. The glomerulus itself needs a very, very, very high blood pressure to allow for passive diffusion to push all of the electrolytes and all of the water into the Bowman's capsule and the convoluted tubes. This high blood pressure is one of the reasons why you need to make sure that you monitor how much glucose is in your blood. Because when you go to squeeze that blood, if there's too much glucose in it, it can cut through the glomerulus. And when you cut through the glomerulus, you then leave opportunities for A, glucose to be in your urine, and B, eventually enough damage to occur to the glomerulus that blood just starts entering the urine. Obviously, it's not a good idea to be peeing blood. So glucose control, because of the nature of this very sharp molecule, is incredibly important to overall glomerulus function because of that incredibly high blood pressure. Now, that, I think, introduces us to our major organs in the nephron, or the major components of the nephron. So now let's take a look at your book's drawing. Now, this is the component. This next step is the component that I expect you to be able to recapitulate with your own drawing. So, of course, we're going to look at it in 3 different ways. First, this very detailed drawing from your book. Then, I'm going to turn this drawing into a cartoon, so that way we can make sure to understand what's going on. And then lastly, we'll look at a schematic of how specifically the loop of Henle works. So, let's get to it. First and foremost, the glomerulus. Let's grab our highlighter because that's going to be important. The glomerulus is going to be filtering solutes from the blood. So it's grabbing everything. Both the good and the bad. You throw the baby out with the bathwater, then you go out to where you threw things out and grab the baby to bring them back in. That's the basic idea behind the nephron. Everything leaves, then you go back and you pick up what you want. The first step of picking up, and as we mentioned before, the glomerulus is doing that filtering by pressure. So it's physically squeezing the blood like a sponge. Next up, the proximal convoluted tube is reabsorbing ions, water, and nutrients while removing further toxins and adjusting pH. So that was a lot and kind of quickly, so let's go through the caption for this figure and read this a little bit more closely. So the proximal convoluted tube, reabsorbing ions, water, and nutrients from the filtrate into the interstitial fluid, and actively transport toxins and drugs from the interstitial fluid and into the filtrate. So you are actively moving toxins and drugs. So anything that's left in the blood after leaving the glomerulus, because you're not going to squeeze everything out, right? The blood is running around these convoluted tubules. You remember the efferent arteriole? It's still dancing around the tubules. And the reason why it's doing all that dancing is so that way it can continue to pass in things that maybe didn't get through. Any toxins that are there or any drugs that are there, so that way you can pee them out. Now, it's also adjusting the blood's pH by selectively secreting ammonia, NH3. This is one of the defining features of what the proximal convoluted tube is doing, is it's a pH regulator. It secretes NH3. The NH3 will then react with the H pluses in the filtrate to form ammonium. So this is the first step on your way to making urea within your urinary bladder. Now, we know that the kidney has its major role in that excretion of NH3 molecules, of producing urea, and that the source of your ammonium or your nitrogenous wastes is actually protein metabolism. So the major function that we oftentimes associate with the kidney is actually performed by that first proximal convoluted tube. It's secreting ammonia to regulate our blood's pH. And the reason why there might be too much ammonia in your blood is because you are breaking down proteins. Regular function, got to make sure to balance pH to maintain homeostasis. So, let's highlight this last portion here so that way we don't forget it. Also adjusts blood pH by selectively secreting ammonia into the filtrate where it forms ammonium. The more acidic the filtrate, the more ammonium is secreted. So you are actually releasing more ammonium if your filtrate is highly acidic. So if the solutes that got squeezed out of the glomerulus had a lot of H+, you will remove more NH3 to balance things out. From there, reminding ourselves of the journey, step 1, out of the glomerulus, step 2, from the glomerulus to the proximal convoluted tube, and then you take this journey down the descending loop of Henle. And you'll notice that there is a wide portion and then a thin portion. And the same is true of the ascending. There's another wide portion up top. More about that in a second. But the descending loop of Henle is characterized by one group of proteins, the aquaporins. They allow water to pass back from the filtrate into our interstitial fluid, back into the body. We want to hold on to our water, depending on how much water you have in your bloodstream, and the descending loop of Henle is doing that. So when you consider when you think of the descending loop of Henle, always think of water reabsorption. So the descending loop of Henle is lined with aquaporins to allow water back in. And remember, we'll revisit this at the end of the lecture, but the amount of aquaporins there are actually hormonally controlled. If your body has a lot of water and not a lot of electrolytes, your water may be more dilute, less yellow pee. If you haven't had a drink of water for a very long time, more likely that water needs to be retained and your urine will be much more concentrated. After the descending loop of Henle, we then round the curve and go up the ascending loop of Henle. There will be no more aquaporins on this wall because instead we are reabsorbing sodium and chlorine ions back into the body. Obviously, our bodies love sodium. We need a lot of these ions. Just remember our neuron function. And in order to keep these, because a lot of them are squeezed out in the glomerulus, we need to selectively bring them back. So those specific transporters are present in the ascending loop of Henle. So in the thin part, you've got them diffusing back into the interstitial fluid, and then in the thick part, these same ions are actively transported. So down at the thin, it's diffusion, and then up at the thicker, it's active transport. But altogether, the entire ascending loop of Henle has this reabsorb electrolyte function. And as you go up the ascending loop of Henle, you then reach the distal tubule, the distal convoluted tubule, which is going to selectively secrete and absorb different ions. So here, these are the most flexible, meaning that certain things are let go, certain things are brought back in. If you need more positive ions, your body will go back or your distal convoluted tubule will go back and grab these particular positive ions. If you need more negative ions, it's looking to grab these back. But this is the most flexible component, so that way your blood's pH and electrolyte balances are appropriate. In particular, the ions that are most commonly reabsorbed by the distal convoluted tubule are potassium and hydrogen protons. They are selectively secreted into the filtrate, while sodium, chlorine, and bicarbonate ions are reabsorbed. So, when you think of the distal convoluted tubule, you should be considering KH out and NaCl, and then bicarbonate in. So, for our online quiz, it will be important to keep track of all of these different ions. You'll notice that the vocabulary list is somewhat shorter for this lecture because there's so much more emphasis put on keeping track of which ions are moved during each component of our nephron filtration. Now, after your pH has been balanced and electrolytes have been balanced, you then reach your collecting duct to reabsorb solutes and water from the filtrate, forming our dilute urine. Whew, what an adventure. This drawing this professional schematic is something that I will repeat in a slight cartoon fashion now. Okay, so let's get to our new page and let's do it. So, we of course begin with our glomerulus and the Bowman's capsule. We then go to the proximal convoluted tube, descending loop of Henle, ascending loop of Henle, distal convoluted tube, now into your collecting duct. So that's the anatomy of the thing. So let's go ahead and label everything. You have the glomerulus, the proximal convoluted tube, the descending loop of Henle, the ascending loop of Henle, and the distal convoluted tube, leading to your collecting duct. So a little bit of acronyms there to help simplify this drawing. The role of each of these is going to be shown in red. The glomerulus is going to filter out all the good and the bad to generate your filtrate. The filtrate will then travel to the proximal convoluted tube. In the proximal convoluted tube, ions are being reabsorbed. Water's being reabsorbed. So you've got your aquaporins. Nutrients are being reabsorbed. Toxins are being placed in, and pH is being adjusted. That pH adjustment is the unique component here, so it's what we list. The pH adjustment happens by the secretion of NH3 into the tube from the surrounding arterioles. So, pH is also something I'm going to list. On your descending loop of Henle, we have aquaporins. These are causing water to leave. So at the bottom of the descending loop of Henle, we actually get the most concentrated filtrate during the entire journey. So here, it is the most concentrated filtrate. So I like to draw out some numbers to help illustrate this. Let's go ahead and say that at the beginning, when you squeeze out of the glomerulus, let's put some random numbers to this. Let's do it in hot pink so that way you don't confuse this. Let's say that there are something like 200 parts per million. Going through the proximal convoluted tube, you're actually adding in the NH3. This will up it to something like 300 parts per million by the time that you reach the top of the descending loop of Henle. As it's traveling down the descending loop of Henle, you are pulling out water. So you're going from 300 to 700 to 1,000 parts per million to at the very base. It is extremely concentrated. All the water that's extra is now gone. But there are ions in there that you want to reabsorb. And anyway, that high concentration, that would be very, very concentrated urine. So we need to dilute it down a little bit here for storage purposes. So going up the ascending loop of Henle, we are starting to reabsorb some of those important ions. Now, those ions are NaCl, good old salt coming in from the ascending loop of Henle. As you are pulling out more and more salt, you're actually becoming less and less concentrated because you're pulling out so much salt. The numbers on this might be same position, about 1,000. And keep in mind that natural or effortless diffusion is governing it for the thin part. But then as you go to the thicker part or the higher up on the ascending loop of Henle, now you're using energy to pump out salt. And oh boy, are you pumping out a lot of it. You're going up to 500, and at the very base, you'll be back down to 100 parts per million. And now that 100 parts per million is much closer to exactly the things that you do not want. The very last step in our reabsorption pathway here is the distal convoluted tube, where now you've got some things coming in and some things coming out. Of course, those things coming out are, potassium and hydrogen protons. And then things coming back into the body are more salt and bicarbonate. Let's make this a little neater. My neatness always can get all the help it can get. So those things coming back into the body, potassium and hydrogen going out. And then the collecting duct goes off to the ureter. And so this is my little cartoon. Glomerulus filters out all. The filtrate then goes to the proximal convoluted tube, where pH is regulated by the addition of NH3 into the filtrate. Then it goes down the descending loop of Henle, where aquaporins pull out water, making it extremely concentrated at the very bottom. Salt is reabsorbed in the ascending loop of Henle. And then potassium and hydrogen are pumped back out, and the distal during the distal convoluted tube, and sodium chlorine and bicarbonate. I guess my notation here of in versus out is not all that helpful. Let me show that this stuff is going into the duct and this stuff is going back to the body. Because you are trying to get more stuff going back to the body, water back to the body, and this goes into the filtrate. This goes into the filtrate. So draw this a couple times, just like how you did with cellular respiration, and make sure that you master it, right? Very important concept with a lot of moving parts. The diffusion gradient, and to help illustrate these concentrations here, I like to show this image from your book as to when diffusion is governing things and when you have to be actively pumping. When things are so highly concentrated down here at the bottom, you can actually get free diffusion, moving the NaCl back into the interstitial fluid. But whereas up higher, you have still a lower concentration of things, you're going to need to actively. So this place here is the switch from passive transport to active transport. And that's why the kidneys are using up so much energy. So much energy. It's a stepwise change to maximize your diffusion for free and use active transport or diffusion for cost because it's going against the concentration gradient. The very last thing that we need to cover are the different forms of nitrogenous wastes. Many invertebrates and aquatic species stick to ammonia, NH3. Mammals still utilize some water in their urine and will excrete urea. Birds are extremely efficient with their water. In fact, many insects, land snails, and avians, as well as a lot of reptiles, will excrete a white compound called uric acid. So this is a much more concentrated form that actually comes out a little bit solid. You'll notice that bird poop is not just poop. The white stuff is the bird pee. It is much more solid than our urine because they are retaining a lot more of their water. And then lastly, I just want to remind you that there are certain hormones that will play a role with our kidneys. In the past, we've talked about ADH, or antidiuretic hormone, and aldosterone. So please keep those in mind. But hopefully this gives you a good idea of the complexity and beauty that is involved in kidney function. I promise you it will make sense when you keep track of yourself as a filtrate. So pretend for a second that you were one of these molecules that gets pushed out in the filtrate. What's happening to you along your entire journey through the loop of Henle? If my drawing is a little bit complicated, your book's drawing is much more neat and compact. But know that kidney function is incredibly important and is a good barometer of your understanding of biology to this point. Plus, it'll get its own essay question on the final, right? Hope you enjoyed this.